in highly in highly seemingly random but actually quite orchestrated manner, bringing them together in a derivative chromosome at the end. And obviously, so we're using short read sequencing, we are using paired end sequencing and classical structural variant callers. But the promise of long read sequencing is really to span multiple breakpoints, which can, for example, by local assembly, give us much higher resolution of these complex rearrangements. So I want to start out with an example, and Wolfgang already introduced the disease, so the hematopoietic system and the process of getting blood cells, which is quite tedious. And for a computational person, it's still a little bit interesting and funny that it's called complex karyotypes when there are more than 3 rearrangements, really looking at the genomes by karyotyping. But it works in the clinic, so it stratifies aggressive disease. So that's what people are still doing. And we've been applying long read sequencing to complex karyotypes, and the process is the bone marrow aspirate and FACS sorting. And then we're doing the classical karyotype. We're also doing actually Illumina sequencing, deep sequencing, and then Oxford nanopore long read sequencing to try to resolve these complex karyotypes into more fine-grained structures and identify potential new mechanisms. Here's just some examples of what actually looks like sort of real data from patients. What are we getting? So one of the critical issues with nanopore sequencing is that you need quite a lot of DNA, as you probably will know. We can go down to 0.5 microgram. That's the lowest limit right now, unless you need PCR amplification. So we're getting an average of 30x coverage from the cohort and quite nice output. Here you see 2 different examples. With an to the one side is what we see on average is an N50 of 15 to 20 kb, and then we have a few cases with very long reach, much higher N50. And that's really based on the integrity of the DNA you get, and there's not much we can do about it because these samples obviously processed through FACS sorting. And we can also assess the coverage, and I'm just showing here example of high coverage samples.
And in green, a sample with partly degraded DNA and much lower average coverage. And so we used an ensemble approach for structural variant calling. So we used Minimap for alignment and then 5 different structural variant callers, merging and a series of different heuristics for filtering after merging. And here's a circus plot showing the raw structural variant callers finding more than 50,000 SVs in the genome, and obviously lots of false positives here. And I will jump over the long series of filtering approaches we used, because at the end what we get after both using germline structural variants and panel of normals, we drop by orders of magnitude, and now we get actually structural variant call sets that we can use.
And that is really using aggressive filtering with population calls and sort of heuristic filtering of the structural variants using all these callers. So I want to give you an example of a clinically relevant and highly challenging variant in acute myeloid leukemia. So this is the gene called FLT3. So it's an internal tandem duplication of FLT3. It's highly recurrent and it's extremely challenging. So it's a proto-oncogene, the FLT3. It's a biomarker for aggressive disease. It's actually required for the diagnostics of acute myeloid leukemia. It really causes this autophosphorylation and constitutive activation of the gene. You see that here it's it's highly recurrent in AML, and you can detect it by tedious PCR methods. And what has really been the challenge is that I think there's 4, 5, 6 different specific variant callers for Illumina sequencing just to identify FLT3. It's a duplicated region of a highly repetitive region, so classical issue. And so we benchmarked it with 1000x panel sequencing, Illumina sequencing, and together with Oxford nanopore sequencing. And here I'm just showing you from samples where we do identify both the Illumina. So you see here the VAF, the allele frequency, we see the first patient here, it has almost a VAF of 1, meaning there's probably an LOH here region, the other region is lost. So the different variant callers for Oxford nanopore actually really agrees very nicely on the allele frequency of this variant, whereas the Illumina sequencing goes from everything from a VAF of 0 to 0.5, and the VAF is clinically very important to know whether this is a clonal or subclonal mutation. The same goes for the second patient. Also, essentially here, all the nanopore callers based on 30x agree on a VAF around 50%, whereas the 1000x Illumina sequencing goes again from everything from 0 to 50%. And the final one, which is the longest, this was 130 base pair internal channel duplication. So here the Oxford nanopore callers was a little bit less agreeing, but was essentially not called at all by any Illumina callers. So the longer it gets, the more difficult, because they say actually it's longer than Illumina reads, and then it just doesn't call it. So that was very promising. This is how it looks like. One of the examples here, FLT3 detected with nanopore sequencing. Essentially see it as this nice insertion here between. Essentially the region is copied and inserted right next to the region. If you look at the sequence context, it's just a perfect nightmare for a short read caller. And we find it with quite high confidence. So that's something, an example of using nanopore sequencing, which is obviously interesting, but also clinically highly relevant to use in the diagnostics of this FLT3 sample. So the clinicians kept on telling us, we need to find FLT3. We just need it for our algorithm to prognosticate the patients. Okay, so I want to now turn to more research-based analysis, where we're going from simple to two-head to highly complex patterns of rearrangements in genomes. So a part of mutation process in cancer we're quite interested is in the two-hit process of biligand activation of cancer genes, originally proposed by Knudsen, the two-hit process whereby a gene, tumor suppressor gene, needs 2 mutations to be inactivated. So that's quite classical, and that's been the mainstay for many years to identify these tumor suppressor genes. We actually find it can be much more complicated. So this is again analysis we did a couple of years ago, where we profiled a large cohort of cancer genomes, found p53 to be the by far most recurrently bilig inactivated gene. And by that method, we essentially identified that it was typically a copy number loss and a mutation. That was it. So classical two-hit approach for p53. And again, p53 is mutated in roughly 50% of cancers. So actually what we found when analyzing some of the, again, AML samples here, we found actually a more complex pattern here. So here we see p53 sitting right here. There's a clear copy number loss at p53. But there's actually also a loss of the second allele. You see that as a slight drop here in the middle of the gene. So when you use long-read sequencing for this sample here, we actually found a quite complex pattern whereby one end of the p53 is juxtaposed to chromosome 5, and the other end is also. So essentially splitting off p53 in 2 parts, bringing it together in chromosome 5. And you can see in the reads below, this is portraying each nanopore read here, that one end is going to chromosome 5, the other one is rearranging. So generating a highly complex pattern, again, inactivating p53 in a biallelic manner. So we also been working a lot on prostate cancer. I'm just bringing this up because we're seeing this pattern recurrently across many different cancers now with this biallelic activation. So prostate cancer is frequently driven by a mechanism called chromoplexy, which I introduced in the beginning. So it's essentially typically a hormone-driven type of arrangement, whereby multiple different chromosomes are stitched together in very complex patterns. And you see the depiction here to the right, whereby we can identify using short-read sequencing these rearrangements. And there are 2 different types of chromoplexy. It doesn't really matter for this talk here how to identify them, but these are occurring in approximately 30 to 40% of prostate cancer genomes. So we found this using our biallelic activation methodology we developed, a really fascinating pattern of inactivating of p53. So again, here you see these arches here, structural variants connecting p53, and you can see a copy number loss of p53. And what we found again is p53 is disrupted here. It breaks one allele of p53, bringing it close to chromosome 21. And what's quite fascinating is that the other part, again, the other part of p53 is now juxtaposed next to CDK12. So CDK12 is another important cancer gene on the same chromosome as p53. And we look at it here, again, you have p53 broken in two parts. One is bridging, so the 5' end is going to chromosome 21. The other one is breaking CDK12 here on the same chromosome. And when we're analyzing a large cohort of prostate cancer patients, we see CDK12 is causing tandem duplicator phenotype. So the red bars here, these are tandem duplications. As you see those red dots below here, these are patients with CDK12 biligand activation by this mechanism we're finding here that causes essentially small tandem duplications. And you basically just look at the coverage plot of these samples, you can almost kind of visually spot that this looks like a CDK12 inactivated genome. So essentially we have a point mutation of one allele of CDK12 and an inactivating breakpoint of the other. So quite fascinating, the same cancer genome, the same chromosome also disrupts BRCA1. So that's also situated in chromosome 21. So here we see yet another breakpoint in the same complex pattern here. It's one event happening probably in the single cell cycle, juxtaposing chromosome 21 and 16 and BRCA1 as you see here. So essentially, what we're getting at is we can resolve this really complex pattern. Again, this is a pattern we're seeing recurrently of disrupting of p53. So this is using partly locally assembly, the long reads and the short reads together. We're seeing this is our, of course, our model that we have one allele where we have losses of BRCA1 of p53, single allele, and we have a second highly complex pattern which involves 5 different chromosomes bridging together, p53, CDK12, and BRCA1 in a highly complex manner, ending up with, in a single event, biligand activation of 3 tumor-suppressor genes, ending up in this pattern here. By the end, again, you have loss and break and loss and mutations in a single event. And I think the long read sequencing really helped. The local assembly of the long reads really helped to resolve this. Again, this is something which is partly manual, so this is something we are working hard to optimize. And I acknowledge the PhD student who made this. This was not a 5-minute work, but she was also excited to actually try to resolve this, which was in the beginning just complex, as we say in the leukemia field. Okay, so I just want to end here with a summary. So nanopore-based structural variant calls can identify clinically highly relevant variants such as this FLT3. And what we're finding really useful right now is we need to combine both short read and long read sequencing. That really improves the calling of complex SVs. I showed you just one example of chromoplexy, but we're seeing this pattern recurrently again and again, where, for example, especially p53 seems to be disrupted not just by 2 loss, but basically disrupting in very complex patterns, leaving sort of the historical view of just two copy number loss a little bit oversimplistic. And especially these multi-breakpoint long reads, I think these are the ones that really helped us solve these events here. I didn't show our huge efforts in filtering. I just showed the circus plot, which goes from chaos to something you can work with. And what we really need, I was very excited about the PacBio resource yesterday. I mentioned this Colors DB, that we really need for Oxford Nanopore data as well. And I mentioned it to Nanopore folks many times. So this would really help a lot because we need that for both for germline, but also somatic, because there's technical artifacts and then there's the germline variants, and both contribute when we're doing cancer genomes to our false positive rate. So I'll end here and thank the people involved, especially Andre, Jakob, and Daria, and the collaborators in PBCG and the Blood Cancer Consortium and funders, and you for your attention. Thank you.
Thank you very much. I couldn't agree more that we need such a database. It's data.
Yeah.
So I was wondering, you probably also worked with PE material in some of your studies, and there are these rumors that they can generate very long reads. So what is your personal experience?
Yeah. So obviously FFP is where all the biobank material is, probably 98-99% of all tumor material. Some of it is really just the greatest single strand, 180 nucleotide, right? And that of course is useless, but some of it is quite high, long range, yes. So we are able to identify quite long sequencing reads up to, I think we're getting 10-20 kb of nanopore from FFP. So I think that's definitely, it's very useful, but you need to identify them first, right? So what we just put in a gel and check whether there's long range material.
We have two short questions. The first one is, do you think that in this reference data set released, the methodology will play a role in defining this technique, for example, next answer, some value, or if it's somewhere?
Yeah, the question is the method, right? And that's also what we're finding, that so Snipples is probably the one that performs the best right now, but it's definitely not perfect. I think there's also something with the alignment, which also needs improvement. So we typically use Minimap2 with default parameters, but I think there's room for optimization. And then of course, if it becomes n squared number of combination, the more you add. But I think we actually we need to identify a couple of variant callers that I mean, it's still a very active field, and we just heard Manuel working on another variant caller, right? I mean, especially the indels are a huge issue. So yeah, I would agree, we can't rely on single caller.
The second question is, does this method also apply on relatively heterogeneous tumor, and where like this threshold in similar, for example, we can detect less than 10 or 20% phenotype in your genome?
So the question was, do you mean in population or mosaicism or clonality? Right. Yeah. So we're definitely struggling with a low subclonal SV. So the sensitivity drops quite dramatically. I think someone showed a very nice sensitivity and specificity for germline SVs, but these are all there for 50%. And Manuel just showed an example. This is what we're seeing as well. And especially in cancer genomes, I don't think 30x is probably enough to find subclonal structural variants. We have a couple of ONT with 60-70x, and we've downsampled them. We haven't analyzed them thoroughly to kind of figure out kind of a VAF sensitivity spectrum. And again, that's also very different for the indels, which are a huge issue, right? I think the duplex read sequencing will improve it. We're just starting with that as well. But yeah, so we're mostly working with structural variants, and there it's a little bit easier than the indels.
Thank you very much for this amazing work. What you do. I actually wanted to ask a question on the frequency. So whenever you have daytime, then you range.
Yeah, absolutely. And I think that's why we're quite far from the germline benchmark data, because many of these are indeed subclonal, and I guess for the Illumina sequencing, we know quite well what our sensitivity is. We just don't know for long read sequencing. That's also why it's an exciting field, right? Because it's so fastly evolving, and there's always a new update if you check. So that's exciting, but it also means that it is a moving target. But definitely low subclonal events are a huge issue also with 30x, because it's a numbers game.
certain version. Last question. So, thanks a lot. Thanks to all the speakers of this very great session. I